There's something really fundamental, almost sort of wondrous about how life replicates itself, isn't there?
Absolutely, how just one single cell becomes two.
Oh,
Identical
and it
copies.
underpins everything. I mean, how you grew, how your skin heals after a cut. Yeah.
It really is the cellular blueprint in action, this like incredibly precise process happening inside you billions of times right now.
That precision. Yeah, that's exactly what we're diving into today. We're going to unpack the mechanics of cell division.
Focusing on mitosis and the whole cell cycle.
Exactly.
But
And
right.
we're pulling this straight from the articles and research you shared, the material laying out the key stages and, why it's all so important.
Yeah, our mission basically is to figure out from these sources not just that one cell becomes two, but how,
How it makes sure those two cells are genetically identical.
right? And why that identity is so crucial for, well, most of your body.
Yeah.
We'll trace that step-by-step journey the sources describe.
Okay. So, at its heart, we're talking about mitosis, a specific kind of cell division. The goal: two genetically identical daughter cells, diploid ones.
It's essentially cloning at the cellular level.
And the sources are pretty clear on why cells need to do this. Growth is the big one, right?
Yeah, the obvious one. You need more cells to get bigger. Simple as that.
But also repair. That seems just as critical.
Oh, definitely. When tissues get damaged, you scrape your knee. Whatever mitosis is what replaces those injured or dead cells
Your body's little renovation crew working away.
constantly. And interestingly, the material points out that for simpler organisms like bacteria, some plants,
Mm-hmm.
this is how they reproduce.
Asexual reproduction.
Exactly.
Yeah.
One parent cell just divides by mitosis, creates genetically identical offspring.
Yeah.
No partner needed.
And the common thread here, the thing the sources really hammer home, is that essential outcome. The new cells have to be genetically identical to the parent.
Yeah, identical. Any slip-ups could cause big problems down the line.
Right. And getting that perfect copy, distributing it correctly, it's not just one quick event. The sources talk about it as a cycle.
The cell cycle,
Well,
a series of, predictable stages a cell goes through as it grows and gets ready to divide.
okay, so the overall cycle, the material seems to break it down in a couple of slightly different ways.
Yeah, one source lists it as like growth, then making DNA, then more growth and checking for errors,
Mm-hmm.
then mitosis, and finally the cytoplasm splitting.
Okay.
Another way you shared frames it more broadly: interphase, then mitosis, then cytokinesis.
Which is basically the same journey, just different chapter headings, sort of.
Pretty much. And
Yeah.
interphase is, well, it's critical. It's the longest phase.
Yeah.
The cell isn't actually dividing yet, but it's working hard.
It's prepping, like a pregame warm-up.
Exactly that. An elaborate warm-up.
And the prep involves some heavy lifting, right? The sources say the DNA, the chromosomes, copies itself during interphase.
That's the absolute key task here, replication. So each chromosome ends up consisting of two identical sister chromatids joined together, a perfect duplicate set.
Got it. And the cell itself gets bigger, too.
Oh yeah, it bulks up, grows larger, makes more organelles, mitochondria, ribosomes, all the stuff the two new cells will need.
And the material mentions something about how the chromosomes look during this phase.
Right, they're long and stringy, decondensed is the term. You can't really see them clearly under a regular microscope at this stage.
Like tangled threads just doing their normal job before being packaged up.
Exactly. Okay, so once interphase is done, DNA copied. Cell bigger, it's ready for the main event, mitosis itself.
And your sources list the four core stages: prophase, metaphase, anaphase, and telophase.
Yep, in that order. Following interphase, leading up to the final split, which is cytokinesis.
So prophase kicks things off.
It does. It's pretty dramatic, actually.
Yeah.
Those spread out chromosomes, each with its two sister chromatids, start to condense.
They coil up tightly, become visible,
Precisely. Thick enough to see under a microscope. It's like winding that thread onto spools, like you said.
and the nuclear membrane dissolves.
Yeah, the membrane around the nucleus breaks down, disappears, clears the stage, basically, and spindle fibers start to form.
These structural things from centrioles in animal cells.
Right. They emerge and start moving towards opposite ends, the poles of the cell. These fibers are the machinery that'll do the separating.
Okay, so prophase sets the stage. Then comes metaphase.
Metaphase, M for middle. It's quite striking visually.
The chromosomes line up.
Exactly. Each one, still made of two sister chromatids, lines up perfectly along the cell's equator, the middle line,
Like they're queuing up very neatly.
very neatly. And those spindle fibers we mentioned, they attach to the centromeres.
The point where the sister chromatids are joined.
Yep. This perfect alignment, the sources call the metaphase plate, is crucial. It ensures each new cell gets one copy of every single chromosome.
Like sorting everything perfectly before delivery?
Couldn't have said it better. And then comes anaphase, O for warrior, away or apart.
This is the separation part.
This is a separation. It's fast, kind of furious. The spindle fibers shorten, contract.
And pull the sister chromatids apart.
Exactly. They yank them apart. And since they were identical copies, they get pulled to opposite poles of the cell.
And once they're separated, they're considered individual chromosomes again.
That's right, because they're now independent units heading to different destinations. You've successfully separated the identical sets.
Okay, makes sense. So chromosomes are at opposite ends. What's next? Telophase.
Telophase. T for 2, perhaps. The cell starts to sort of reverse what happened in prophase.
How so?
New nuclear membranes start forming around each group of chromosomes at the poles. 2 new nuclei are being built.
Ah, okay. Containing those complete identical sets of chromosomes.
Exactly. And the chromosomes themselves start to relax, to decondense, become less visible again.
So the nuclear division part is basically done by the end of telophase, but the cell isn't quite 2 yet.
Not quite. That final physical separation is cytokinesis.
Splitting the cytoplasm.
Right. The sources describe the cell membrane pinching inwards, especially in animal cells, like pulling a drawstring.
Creating a furrow.
Yeah, a cleavage furrow that gets deeper and deeper until, pop, the cytoplasm divides, and you have 2 totally separate daughter cells.
And the result, which the material keeps stressing.
2 genetically identical daughter cells.
Diploid body cells, somatic cells, each one with the exact same number of chromosomes as the parent cell had.
Absolutely. So for us humans, if the parent cell had 23 pairs, each new daughter cell gets 23 pairs. The fidelity is incredible.
And the sources even say they're clones, right? Every base pair of DNA is meant to be identical.
That's the goal. It really makes you appreciate the precision needed at every single step.
It's kind of mind-boggling.
It is. The cellular machinery is astoundingly accurate. Most of the time, anyway.
So let's sum up why this matters so much based on the sources. 3 main pillars, it seems.
Sounds right. First up, growth.
Simple enough. More cells means the organism gets bigger, tissues expand, organs develop, all thanks to mitosis.
Second, repair.
Replacing cells lost to wear and tear, injury, illness, like we said, healing cuts, but also constantly replacing things like blood cells, skin cells.
Your gut lining gets replaced very quickly, all through mitosis.
And third, that point about asexual reproduction.
Yeah, for many single-celled organisms, and even some multicellular ones, it's how yeast buds, how a plant cutting can grow roots. It's cloning for survival.
Okay. And it's worth repeating, you mentioned this earlier, this is all about body cells, somatic cells.
Strictly, sex cells, sperm and egg, the gametes, they're made differently. Through meiosis, different process, different goal.
Right. The material also had some specifics, like confirming humans have 23 pairs of chromosomes in these diploid cells.
Yeah, grounding it in our own biology.
And there was that interesting little formula, too, for estimating time.
Oh, yeah, for people actually, looking at cells dividing under a microscope, if you want to estimate how long a cell spends in, say, metaphase,
You count the cells you see in metaphase.
divide by the total number of cells you're observing.
And multiply by the total time for the whole cell cycle, if you know it.
Exactly. Gives you a rough estimate for the duration of that phase in that particular tissue or culture.
That really brings it home that this isn't just abstract theory. It's a measurable biological event.
Happening constantly.
And when you think about it, understanding this process is just. It's fundamental to understanding how your own body built itself, how it maintains itself second by second.
It's the engine of continuity at the cellular level, making sure perfect replacements are ready when old cells wear out or get damaged.
So, okay, quick recap. We've gone through the whole cell cycle journey. Interphase, the big prep stage, copying DNA, bulking up.
Then the 4 acts of mitosis itself. Prophase condensing chromosomes, nucleus dissolving. Metaphase lining up in the middle. Anaphase pulling the identical copies apart. Telophase forming 2 new nuclei at the ends.
And finally, cytokinesis. The actual split, resulting in 2 perfect genetically identical daughter cells.
The key takeaway from all the sources. Mitosis is about making exact genetic duplicates. For growth, for repair, for asexual reproduction, it's all about fidelity.
Right, precision. So, given how incredibly precise this whole thing needs to be at every single step to ensure that identical genetic information gets passed on, here's something the sources kind of hint at, a thought for you, the listener, to chew on.
What happens when it goes wrong?
Exactly. What happens when this incredibly controlled process with all its checks and balances fundamentally breaks down? When cell division becomes unregulated?
What are the potential consequences when the system built for perfect copies starts making mistakes or just divides without the proper controls? Something to think about.